Hello everyone, welcome to the Insights of Viral Microwave series. Today I will discuss about bacterial metabolism part 3, phototrophy part 2. In this session I am going to cover about oxygenic photosynthesis, whole process, photosystem 1 and photosystem 2. So let's start with the session. So this content is a part of bacterial metabolism MI 302 Unit 3 as per the Gujarat University syllabus. So let's start with oxygenic photosynthesis. Before starting this, I just want to explain about oxygenic photosynthesis follows two photosystems, photosystem 1 and photosystem 2. Now, photosystem 1 absorb longer wavelength light and funnel the energy to a reaction center, chlorophyll, a pair called P700. Now, the term P700 signifies that this molecule must effectively absorb light at a wavelength of 700 nanometer. Now, photosystem 2 traps the light at shorter wavelength and transfer its energy to the reaction center, chlorophyll pair P680. Means this particular photosystem 2 is going to trap the light at a wavelength of 608. Now, photosystem 1 can function independently, but depends on the organism's need. PS1 and PS2 both simultaneously work at certain times. So let's start with the detailed presentation. Okay, so when light is absorbed by reaction center, chlorophyll pair in a photosystem to chlorophyll molecules change from having a very positive standard reduction potential to a very negative standard reduction potential. So two photosystem working together going to generate ATP and NADPH, and oxygen is going to be released in case of oxygenic photosynthesis. Now, as I had explained you about the positive standard reduction potential and a negative standard reduction potential, that will I explain you in detail. But before that, I just want to explain you about the photosystems. So from this figure, I hope you can see that the absorption of light causes the reaction center chlorophylls to become excellent electron donor. Now, I just want to explain you this. See, this is a photon absorbed by the reaction center, right? And at this reaction center is become energetic, means it is having now very negative reduction potential, and now it is a very excellent electron donor. Now we will first examine the photosystem 2. So I hope you can see that photosystem 2, here we first examine photosystem 2. When it absorbs light, the energized P680, this is P680, will become P680 star. So star means it is energized. Now this energized molecule donates electron to a series of electron carriers that ultimately donate the electrons to P700 of photosystem 1. So now, who are the series of electron carriers? First carrier, second carrier, third, fourth, and by the help of this two, this particular electron which is absorbed by P680 transferred via these electron carriers to P700. Now the flow of electron through P680 associated electron transport chain generates a proton motive force which can be used by ATP synthase to make ATP from ADP and PI. So from this you can see this ADP and PI converted to ATP via ATP synthesis. Now, meantime, photosystem 1. Now this photosystem 1 has also absorbed light and it's energized. P700 has absorbed the light, it has energized to P700, so it become P700 star now, and its energized reaction center chlorophyll P star 700 donate electrons to a short electron transport chain that ultimately transfer electrons to NADP plus yielding NADPH. Now see, this is a electron transport chain of this particular photosystem, and this NADP is converted to NADPH. So by this way, ATP and reducing power has generated. Thus, when both photosystems work together, both ATP and NADPH are produced. Because the electrons flow is in a non-cyclic pattern, this is called non-cyclic electron flow, and the ATP produced is said to be made by non-cyclic photophosphorylation. Now here, cell is having a problem. Okay, so now what type of problem cell is going to face during this? Now see, as you have seen that P700 has absorbed light, P680 has also absorbed light. Now P700 is oxidized. So for the next electron acceptance, it has to be reduced once again. So this electron which is accepted by or absorbed by the P700, that electron is replaced by P680, right? So once again, आपको दोबारा मैं समझाती हूँ. दोनों फोटो सिस्टम मतलब P700 एंड P680 दोनों ने इलेक्ट्रॉन एब्सॉर्ब कर लिया है. 700 वाला जो इलेक्ट्रॉन है, it will going to be replaced by P680. So it is once again reduced, तो अब वो दोबारा एब्सॉर्ब कर पाएगा. Now for P680, it is going to absorb light continuously. Then what about the replacement of that electron which has absorbed by the P680? So here the water molecule will help to replace the electron, as P680 is having a very positive reduction potential as compared to the H2O. So H2O will replace the electron from that P680 reaction center. So for this reaction, oxygen evolving complex enzyme is going to help to complete the reaction, and by this oxygen evolving enzyme, this particular molecule of H2O will going to be split, and because of this reaction, oxygen is going to be released into the atmosphere. So by that way, if the PS1 and PS2 is simultaneously working, then H2O will split and oxygen molecule will be released into the atmosphere. By this way, this particular PS1 and PS2 is working together. So when photon has or light is absorbed by PS2 or P680, immediately the Mn, see here, this is a manganese molecule, and OAC complex is going to be increased. Oxygen evolving complex is going going to increase into the cell, and they are going to work for the splitting of H2O, and it is converted to O2. So by this way, this reaction is going to be happen. Okay, so let's start with the photosystem 1. As we have already discussed about photosystem 2, let's move on photosystem 1. So photosystem 1, same way working as photosystem 2, but photosystem 1 can able to generate ATP if it is functioning only. Then it is able to generate ATP, and ATP generation by photosystem 1 is in a cyclic manner. So the ATP generation is called cyclic photophosphorylation. So now see, as you know that P680 and P700, the electron carriers, or that is electron transport chain, is correlated, right? So by that way, this particular electron carriers are attached with each other. But if photosystem 1 is only functioning, then only ATP is going to be generated by cyclic photophosphorylation. And if both are working, then only ATP and NADPH both are going to be produced. Now let's move on. In which circumstances both the photosystem are going to be work together, and in what kind of circumstances only photosystem 1 will going to work. Now this particular question is depend on the requirement of organism as well as the requirement of dark reaction. So let's move on the dark reaction. Now, sorry. So in case of dark reaction of oxygenic photosynthesis, see, one molecule of carbon dioxide fixation, for the carbon dioxide fixation, one molecule of CO2, you require, cell require around 3 ATP and 2 NADPH. So the ratio of ATP and NADPH is 3:1. Sorry, 3:2. So now around 4 electrons has going to be produced, 2 ATP and 2 NADPH. Now, so for 4 electron, 8 quanta of light is absorbed by the cell. Now, as you know that 3:2 ratio of ATP is S2 NADPH is there. So, around 12 quanta of light is absorbed for the one molecule of CO2 fixation. So, you can see from this, one molecule of CO2, 3 molecule of ATP, 2 NADPH, 2 H plus and 2 H2O converted to carbohydrate plus 3 ADP plus 3 PI plus NADP, and 1/2 O2 is going to be released during the photosystem process, PS1 and PS2. During the CO2 fixation, there is no oxygen is going to be released. So I hope you understand about. So before clearance of this topic, you should be clear about the redox potential. What do you mean by positive redox potential? And what do you mean by negative redox potential? So I just want to clear these words. Positive redox potential means it's like carbon dioxide, CO2. It is not having any hydrogen. So it is having very high or very positive redox potential. And if it is having, for example, methane, CH4, so it is having negative redox potential as compared to the carbon dioxide. So if you want to oxidize particular molecule, then CH4 is having very negative redox potential, so it is easy to go for oxidation, right? Whereas for CO2, if you want to reduce it, then it is required high energy for the reduction. So I hope you clear about positive and negative redox potential. And once again, I just want to clear that P680 is going to replace the P700 electron, and by that way only the next photon is going to be absorbed by P700. And for P680, as the electron is replaced by the H2O, H2O is converted to oxygen, and by that way this particular photosystem is known as oxygenic photosynthesis, and this is done by cyanobacterial species. So I hope you understand about this. Thank you, everyone.